In this video, I am going to talk about glycolysis. Glycolysis is the most fundamental of the metabolic reaction. The glycolysis comes from two words, glyco, which is derived from glucose, and lysis, which means breakdown. So, glycolysis is the breakdown of glucose. So, in the previous video, I talked about the transport of glucose from the extracellular space into the cell. When the cell takes up the glucose, it then breaks down the glucose and it forms pyruvate. That pyruvate is again converted into other molecules such as acetyl CoA. That acetyl CoA serves as an intermediate in citric acid cycle. The glycolysis also yields some energy in the form of ATP. Because it is an energy rich substance, this can be broken down and it can generate energy. Glycolysis also serves as glycolysis also provides many intermediates, and those intermediates can be fed into the citric acid cycle or oxidative phosphorylation. And the acetyl CoA that is made from pyruvate, that acetyl CoA serves as a precursor for the synthesis of fatty acids. So this was some of the significance of glycolysis. Now moving towards the reactions. Glycolysis is composed of 10 reactions. First 3 or 4 reactions, they are the preparatory reactions, and the next reactions are the energy yielding reactions. In this video, I will only talk about the preparatory reactions. The first reaction of glycolysis is the conversion of glucose into glucose 6 phosphate, and this reaction is catalyzed by hexokinase. The name of the enzyme implies that hexo, which is indicating the sugars in the hexose form, and the kinase is the enzyme that adds a phosphate group. So hexokinase adds a phosphate group to the glucose at the sixth carbon and makes it or converts it into the glucose 6 phosphate. This phosphate group comes from the ATP. The terminal phosphate of the ATP is removed and it is then uh, attached or added to the sixth carbon of the glucose, and we get glucose 6 phosphate. This reaction of glycolysis, which is the first reaction, it is an irreversible reaction. So why the cell needs to convert glucose into glucose 6 phosphate? In the previous video, as I mentioned, that glucose is transported from the extracellular space inside into the cell. The cell needs to trap this glucose inside the cell. So in this trapping reaction, the phosphate is added to the glucose. As the phosphate has a negative charge, and the internal leaflet of the cytoplasm is also negatively charged, the charge of the the plasma membrane is negative. So the this force of repulsion between these two molecules, the membrane and the phosphate, it traps the molecule, it causes repulsion, and it traps the glucose inside the cell. The cell is also continuously transporting glucose from the extracellular space inside the cell. So if the glucose is not converted in some way, the store of glucose inside the cell is going to saturate, so the cell cannot further take up glucose from the extracellular space. So cell tags this glucose or modifies this glucose from in the form of glucose 6 phosphate from glucose, so that cell can continue taking up the glucose from the extracellular space. The third thing is that this glucose is inactive. The cell wants to activate it, so it adds a phosphate group to this glucose, and now this glucose becomes activated, and now this glucose can be used in the further reactions of glycolysis. This was the first step of the glycolysis. The second step of glycolysis, in which glucose 6 phosphate is converted into fructose 6 phosphate, it is an isomerization reaction, and this reaction is catalyzed by phosphoglucose isomerase. We cannot appreciate this reaction from the ring structures well. For understanding this reaction, why is this reaction taking place, and the significance of this reaction, we need to have a look on the open chain structures. So glucose 6 phosphate is an aldose sugar, and the carbonyl group, as you can see, is at the first carbon. So in an aldose sugar, the carbonyl group is present at a terminal of the molecule. But in the case of fructose 6 phosphate, this carbonyl group is transported or transferred from the terminal of the molecule to the to the interior of the molecule. So in the glucose 6 phosphate, the carbonyl is at the carbon 1. In the fructose 6 phosphate, the carbonyl is at the carbon 2. What is the significance of this reaction? This reaction, this transfer of carbonyl from the terminal to the interior of the molecule, it transports the reaction center from the terminal of the molecule inside the from the terminal to the interior. This carbonyl, the oxygen of this carbonyl is more electronegative, so it will attract electrons towards itself. So it is going to create a disturbance inside this molecule and in the vicinity of this carbonyl. So it becomes easy to cleave this and this carbon-carbon bond. The carbon-carbon bond between the C3 and C4 becomes easy to cleave. So this is the significance of this reaction. The third reaction of glycolysis is the conversion of fructose 6 phosphate into fructose 1,6 bisphosphate, and this reaction is catalyzed by phosphofructokinase. This is also an irreversible reaction. In this reaction, another phosphate group is added at the carbon 1 of the fructose 6 phosphate and it becomes fructose 1,6 bisphosphate. So these phosphate groups are coming from ATP molecules and they are being added to the reactants, and because it is the preparatory phase, and in the energy yielding phase these uh, phosphates are going to be used for the generating ATP. So till now this pathway has used two ATPs, one ATP in the first reaction and another ATP in this reaction. So the fourth reaction of this pathway is the splitting reaction, in which fructose 1,6 bisphosphate is splitted into two molecules, and both of these molecules are 3 carbon molecules. This reaction is catalyzed by aldolase, and one of the molecule is dihydroxyacetone phosphate and the other is a glyceraldehyde 3 phosphate. Now the only molecule that can go further down the pathway is the glyceraldehyde 3 phosphate. So the dihydroxyacetone phosphate needs to be converted into glyceraldehyde 3 phosphate so that it can also go further down the pathway and yield energy. So you can see that the significance of the second reaction, the transfer of the carbonyl from the terminal to the interior of the molecule, was that to break the bond between the third and the fourth carbon so that the molecule can be split into two. Now the molecules are split into two. The dihydroxyacetone phosphate is going to be converted into glyceraldehyde 3 phosphate by the enzyme Triose phosphate isomerase. It is an isomerization reaction in which this carbonyl is going to move towards the carbon 1. So it is going to move or transported to the terminal carbon and we get glyceraldehyde 3 phosphate. So the only purpose of transporting this carbonyl from the terminal to the interior was to make the bond between the carbon third and carbon 4 weak so that the bond can be cleaved. So the cleavage have been the cleavage has taken place. So this molecule, this carbonyl carbon, can now further move to its previous location, and this molecule glyceraldehyde 3 phosphate can now further continue down the pathway. So why is the 6 carbon molecule split into 3 carbon molecules? It is done to increase the efficiency of this reaction and to increase the energy yielding efficiency of this pathway, because that's why this 6 carbon molecule is split into two 3 carbon molecules So now each of these molecules will continue down the pathway and it will yield energy in the form of ATP. In the next video, I will talk about the energy yielding reactions of the glycolysis. Thanks for watching.